But I do want to talk about genotoxic impurities. And how to handle it. And then what we heard earlier, and again, it's the first time I've heard that they're actually putting holds on people that don't have the SAR like that. But the guidance, and there have been really increased regulatory expectations here. And those of us that have been involved with this since 2006 were very surprised. A lot of clinical holds in phase 2 because genotoxic impurities weren't analyzed. There's really no guidance around at the time. And so it's good that guidances are out there. And where we are right now is that there's draft guidance on that initially came out in 2009. I think the last iteration of this was February last year. It's last guidance for the DNA reactive mutagenic impurities. The difference here is the threshold at which you need to really qualify these impurities. And the threshold, they call it the threshold of toxicological concern. And for mutagenic impurities that damage DNA, there really is no threshold per se, because right now the agency uses what would a linear dose extrapolation to get to a human dose that would cause a certain frequency of cancers. And so right now, the limit that's used here for impurities is really based on several hundred genotoxic compounds that were also rodent carcinogens. So there were compounds that were positive in the Ames test and also had a rodent carcinogenicity study conducted. And that database was looked at, and using a risk assessment factor of 1 cancer in 100,000, assuming a lifetime exposure, looking at those mutagenic compounds, it came up with 0.5 micrograms per day. Again, linear dose extrapolation back to give you a rate of 1 in 100,000. Now, typically in foods, the acceptable risk is 1 in a million. But for pharmaceuticals, where there is a benefit, that risk was set tenfold lower, so tenfold higher rather. So in early development, they have what's called a stage threshold. So in early development, the animals or humans aren't given dose over a lifetime as what a chronic indication. So they're given doses, a single dose, 2 weeks, months of dosing. So the risk assessment is a little bit different, and I'll show a slide on that. But they use early on, it's actually, they go back to a 1 to a million for shorter exposures. So this is a threshold base that's used basically for all impurities that have been shown to be genotoxic. The stage concept, and this is in the draft guidance, shows that I mentioned the 1.5, and this is microgram per day for assuming a lifetime exposure. This is what would be allowed for a genotoxic impurity. If it's a shorter exposure, that number is much higher. It's 120, less than 1 month exposure, 120 micrograms per day. The higher levels are acceptable, again, doing the risk assessment for certain types of conditions that you're treating. Also, I should mention this 1.5, looking at these Ames positive compounds, weren't the most potent Ames positive compounds. There were compounds that were more potent, and these, which would, such as nitrosamines, for example, alpha toxins, they have a higher potency, so they actually have a lower level of threshold with regard to having to qualify. And then, again, all genotoxic impurities that you would have, just like your routine impurities, have to have specifications in your drug substance. So this was pretty good, because there was a time early on when the agency was using 1.5, but it has expanded now. So here's the process for risk and safety assessment for genotoxic impurities. And one, we heard earlier with regard to identifying potential impurities. And I heard one example that Takeda uses with regard to what are the potential ones. Let's get the list. Let's look at them. Let's evaluate whether any of these are going to be potentially genotoxic. Now, there's different ways of doing that. You don't necessarily have to do all those impurities through SAR, but that was an interesting comment. So here, if you understand your process, you have good control of it, you can do a pretty good job in estimating what those impurities are early on. Now, the next piece is looking at the structure and assessing whether that's genotoxic or not. And there's a couple ways of doing it. One, and what a lot of companies do, they look at the available information, alerting functional groups, the literature, to determine whether there's any issues with regard to the molecule that they have. The next level is identify genotoxic or non-genotoxic using these validated structure-activity relationship databases. And there's two types of databases that the agency recommends. One is called a rule-based system, which is your Derek Nexus system, which basically just tells you, you put in your structure and it tells you if one of those moieties has a relationship to another compound, and then it lists what the toxicities are of that compound. So you can do it just with Genotox. It'll tell you if there are any structures there that are in other structures that have been associated with Genotox. It's not a quantitative assessment. It's called a rules-based assessment. And you can actually do a rules-based really by eye. You don't necessarily need a Derek, but Derek helps because it accumulates all the data that's out there. But it's not necessary really to understand a molecule, but the agency does want you to do that. The other is a statistics-based quantitative SAR. There's two types. I mentioned the multi-case, which I actually like, as well as the multi-case construct is their new database. There's several different databases, and they recommend some of the databases because the agency also uses these same databases as we heard. They also use LeadScope. There was both of these were developed in conjunction with the FDA. And so they like it when you do submit information using either one of these databases. Both are good. From this, if you're submitting a report, should really be part of this. A lot of these databases, when you're using these, you will get a lot of false positives can occur from these databases. So it's important to take everything in. One database will tell you this, that's genotoxic, another one will tell you it's not. It's important to take that information, go into the literature, really understand what's driving the positive response in these quantitative databases. And the quantitative SAR is simply, you look at very chemical descriptors for each molecules that are out there, genotoxic, non-genotoxic, they're put together, and then regressions used to, and very statistical assessments are used to determine the likelihood of your compound having a structural similarity with the molecule that's been shown to be positive. In the Ames test, this only uses the Ames test for mutagenicity, because that predicts very well the rodent carcinogenicity study. And one could argue whether the rodent carcinogenicity studies predict human cancer. That's really another story. I do have three slides which talk about substructures, and these are some of the classical Ashby-Tennant alerts here, and LeadScope kind of put these together in a nice fashion, which are very useful, and I go to all the time when I'm looking at a new molecule, which helps quite a bit. Will tell you whether you think you might have a potential problem. And I think if you think you have a potential problem, then you go to the SAR systems and you do it. If you don't think you have a potential problem, I really can't see doing all compounds, even if they're related to drug substance, but it would be difficult to think the agencies are looking for all that. But we could talk maybe afterwards. That's interesting. And I'm not gonna, so those are three, and Benini also has some interest in the structural avoidance as well associated with genotoxicity, mutagenicity primarily. So the next step is you've done your SAR assessment. You have that, and that should be reported in a report along with some sort of evaluation of that. And then you do a hazard evaluation. So the hazard evaluation, really, all that information, what do you conclude, basically? And one of the outcomes of doing the SAR is that it comes out and your assessment is that it's not genotoxic. Well, you're good. Then you can control your impurity as you would a routine impurity. Also, it could be assessed as potentially genotoxic, but can easily be controlled to the threshold level. It's probably not the case because these things are very hard to control, particularly because you have to use different types of detection systems because the levels are so low in your drug substance. And the third outcome, which is typically the more likely outcome, they can be assessed potentially genotoxic, but not practical, or you really don't have the wherewithal initially to purge those from the system, especially during as you're in early development. In this case, what you want to do is you want to conduct an Ames test. Now, the regulations, the way they read right now, which may change because it's draft, regulations read, you can go ahead and put an impurity that's genotoxic into phase 1, phase 2A. The way it reads right now, you don't have to conduct an Ames test. Now, in practice, I don't know if I would do that, and I would assess the genotoxic potential early on and try to deal with it. The way that regulations read right now, you don't have to, and you can test that within at levels above where you are with regard to a threshold, stage threshold that I talked about earlier. So those are the three potential outcomes of your hazard evaluation. The next is Ames testing. Now, Ames testing, you should conduct those with the synthesized impurity, and that's what the agency expects. And it's not a trivial thing sometimes to get up to 1 gram, which sometimes it takes to do these Ames tests of an impurity, especially if it's difficult to make, especially if you can't even make it. So there are some challenges with difficult impurities with regard to assessing the potential for genotoxicity. But if it's negative, you can classify that as routine, and the Ames test trumps any earlier SAR evaluation. So the SAR tells you this is a flaming Ames positive, you do the Ames and it's negative, you're fine. So it's definitely the definitive test. But if you do have Ames positive, you do have to go through, do the monitoring and do the purging to get that down to the level of the threshold at the given stage threshold of toxicological concern. One of the things that people don't do, which is, and there's a good paper out there, I have the reference in the slide deck, is to go back to the cancer potency database. So for example, when they put these, this 1.5 microgram per day, they did base it on the more potent Ames mutagens, but not the very potent, but the more potent. Now, the aromatic amines, as an example, come up a lot. Well, they're not potent Ames mutagen compounds. And so if you were to go into the cancer database of all the aromatic amines that have been tested and have cancer, you can go to the most potent one. Even the most potent one won't be at the 1.5 microgram per day level. So it's a way to qualify an impurity by going in and more looking at the structural class and where it fits in the database and deriving your own threshold for toxicological concern. So this is something that we're hoping gets in to the final M7 guidance, but very important to be able to look at that. So the next step, after you know whether it's an Ames positive or not, you classify these impurities. And the classifications are, for most genotoxic to least, is your class 1, where you have a known genotoxic carcinogen. So it's an impurity that pops up where it's already been tested, and the information is out there whether or not it's it's positive in the 2-year bioassay, at what level is positive. So you can use a compound-specific qualification there. The next is class 2, where you have a genotoxic impurity. Carcinogenicity hasn't been tested, which is typically the case. But and then you're limited to really using the stage approach or some other adjusted threshold of toxicological concern. The next, which is an example that was mentioned earlier about class 3, you have an alerting structure unrelated to the drug substance, and you really don't, but you don't know the genotoxic potential because you haven't done an Ames test. And that's a class 3, and so most people, you do further evaluation, you do an Ames test. But the guidance, again, the draft guidance, says that you can treat these non-genotoxic for phase 1 trials up to 14 days. But I'm hearing something different from the audience that agency is not doing that. So that may come out of the draft guidance. It could very well be. The next is class 4, where you have an alerting structure, and this happens a lot. You have an alerting structure, but it's the same moiety that's in your drug substance, and you did a full Ames test and a full set of toxicology studies, and it's negative. And so you look at that, if you can do that assessment, you can say, well, I've already tested that moiety, therefore I'm going to call this negative, and I don't have to do anything further. So the guidance separates this out, alerting structure that's already in drug substance, you're fine. And then a class 5 where there's no alerting structure or indication of genotoxic potential. So these are the classes that are currently in these draft guidances. So the next step after all this, and safety risk assessment, I mentioned a lot of these already, but just to kind of really summarize, you need to determine what the threshold of toxicological concern is, and that can be based on many different components, whether it's once a lifetime exposure, you have registration, you have to get 1.5. You hire if your dosing duration, if it's an antibiotic, the level is going to be higher because you're only going to be dosing for 14 days or 4 weeks or whatever the indication is. And also lower if you do have a very potent mutagen. You can use, and I want to here's that reference I talked about, which goes back into the carcinogenicity database to develop a threshold of toxicological concern, especially important for moieties that have been shown to be positive but aren't potent mutagens, like the aromatic example. And also, what's the mechanism of action? How can I look at the mechanism of action of what's happening here? For example, we have somebody, Louis mentioned about the synergy, that if it is a molecule that you have an inducer and you're inducing an enzyme, you have to take that into account, the mechanism of action account, when you're setting these thresholds for toxicological concern. The next is multiple genotoxic impurities, and the guidances do handle that. So I just don't have one, I have 5. How do I deal with those? And there are some guidances out there in the draft guidance. Basically, it just increases your threshold of toxicological, or excuse me, decreases your threshold of toxicological concern. It's additive? Yeah. Oh, that's how they're looking at it now? Well, it's interesting because the guidance gives a table, and it's not necessarily additive in that case.
Well, you want to be on the safer side. By making them additive, you're really getting the
sure.
maximum potential, so you're going to be safe.
Yeah, you'll be safe, yeah, additive. But the guidelines do cover that, and there is a stage table. And I thought when I first saw that, that's what I would have thought, additive, but they're not following it. Again, it's a draft guidance, and a lot of things we're talking about in practice, is different. But it's interesting, once guidances get finalized, I typically see the agency backing off on some of the requirements. So guidances are good sometimes, very good with regard to not having to do more than you need to. Again, I mentioned population exposed, very important. Pediatric population. the guidance really, it has to change and actually become even more conservative when you're talking about pediatrics. So that kind of goes the other way. So that's an important factor when you're looking at the safety risk assessment. So the other piece here is, is really evaluating the analytical risk I have. Even though it's a safety assessment, it's important, what can you do? How do you really control for these genotoxic impurities in the systems? There are various ways of doing that. But they can create a lot of problems having these genotoxic impurities in your drug substance. So I mean that's really it. I just want to take hopefully some take-home messages for everybody. One is that when we talk about qualification, you're really looking at the safety of that impurity at the targeted dose and dosing regimen in humans. The qualification needs to be based on sound toxicological principles and practices. There's other ways. For example, we talked about going back into the Carcinogenicity database, talked about in vitro potency, really qualified impurities. The next is to be proactive, use a multidisciplinary approach to control and qualify impurities. Again, get that feedback back early from your potential manufacturing process and the reagents that you're using. Also to, again, to start early as well. And again, early is the earlier the better. The earlier you can understand and handle your process. That's going to help everybody, even the toxicologist. Set threshold in early clinical development that are practical. I mentioned about the consortium that has higher, of course, the thresholds are higher. I would like to see those even be higher early on, but 0.5% is certainly better. So I want to just stress strong communication, understanding between the chemists and toxicologists, of course, key to the success here with regard to qualification.